Good morning. Today we are going to discuss about life feed for sustainable aquaculture. As you know, aquaculture is underwater agriculture where most of the commercially important animals and plants are cultivated for the economic reasons. And in this aquaculture, the most important aspects are seed production and farming. And this seed production and farming is the basis of the aquaculture, whereby we need to produce the seeds. Here the seeds mean the young ones of the fish, or young ones of the prawns, or young ones of the shrimps, and this has to be produced to stockable size, and then it has to be shifted to the ponds where they are grown to the table size, then harvested for marketing purpose. So this seed production is the first step. Unless we have a very quality seed wherein it can be cultured in a high density culture and in a very normal way. So for that reason we need to have a quality seeds, and these quality seeds has to be produced in hatcheries. When we want to produce seeds in the hatcheries, there are two important aspects. One is we should provide a very hygienic condition and good water in which these seeds are produced. And the second one is the young ones of the fish and prawn should be fed with most suitable live feed. Only when we give good live feed, then they will grow and metamorphose normally, leading to the young ones or juveniles which can be stocked in the pond. So seed production is very important, and again I said that for the quality seed production, live feed is very important. So here in this lecture we are going to see the importance of this live feed. And this live feed consists of microbes, microalgae, protozoans, rotifers, cladocerans, copepods, artemia, and worms. Now let me just briefly explain what exactly is the feeding strategy of a fish. So you could see here is the fertilized egg, and this fertilized egg undergoes cleavage, that is division of a single cell into multiple cells, and that process is called as blastulation. And this blastulation leads to the next stage, which is happened to be gastrulation, wherein the mesoderm, endoderm and ectoderm are separated, and it forms into a larva inside the fertilized egg, which after further development, embryonic development, hatches into a hatchling. And this, in the case of fishes, have a endogenous food available in the form of a yolk, and this yolk is enclosed in a yolk sac. Now this larva thrives on this endogenous food, and this may last for few days, and the duration varies from species to species, different species. In some species it is 2 days, in some species it may last for 5 days. So you could see here this yolk is gradually utilized, and at one particular place it is completely exhausted, and as a result now the endogenous or whatever is available inside this larva itself is exhausted for the development of the larva, and then in the next stage now it has to feed. Till this endogenous feeding stage, the mouth is not open. Only when the yolk gets exhausted, the mouth of the larva is open, and it depends upon the external feed, which stage is called as exogenous feeding. Now this exogenous feeding larva requires very specific and the feed which is nutritive with the nutritive value as well as the size spectrum. The mouth size of the larva and the size of the live feed should match. Then only it can feed. So as it feeds, as it grows in length, and also the mouth width also increases, and that leads to the dependence on different sized of feed, and when such feed is available, it becomes a juvenile or a young one, a normal fish, which from the larval stage metamorphoses into the next stage. And once this finally the larva is transformed into a juvenile, it may take 21 days to 30 days. So during this 1 month of development, we need to provide them the proper live feed. Now let us see about this live feed. As I said first, the microbes, particularly the yeast and bacteria, constitutes the live feed. And in the case of yeast, it is happened to be Saccharomyces cerevisiae. It is a baker's yeast. And this baker's is one of the very important feed, and this goes into the feed formulation in most of the feed formulated for both shrimp and fishes. And as far as the bacteria is concerned, it is the Bacillus species, Subtilis, Polyricia and Negaterium are the species commonly used as feed. Now you can see the photographs of the yeast, which is existing in the form of small spherical granules, and this is very small in size. As a result, either it is a shrimp or the fish eat larvae, it can easily feed on them. Likewise, the bacillus is sometimes a rod shaped, and this bacillus is also very small in size, and they have the requisite qualities to become a highly nutritive food for this larvae of fish and shrimps. Now, what is the advantage of yeast? The common baker's yeast, Saccharomyces cerevisiae, are unicellular fungi, and they usually appear spherical and have a diameter of 5 to 10 micrometer, and they are commonly reproduced asexually by a process called budding. Yeast form the ideal food for many zooplankton, which are happen to be again the live feed for the fishes. Yeast forms important ingredient in all feed formulation, as I said earlier, for the larval stages and forming species of prawns and shrimps. A natural sugar which covers the outer cell wall of this Saccharomyces cerevisiae, it improves the gut health of the larval forms. Thereby, it plays a very vital role in the growth and survival of this shrimp larvae or fish larvae. Then the next is happen to be a microalgae. You know that microalgae happen to be unicellular primary producers, are the organisms which are capable of synthesizing the starch from making use of the carbon dioxide and the solar energy when there is a chlorophyll, and this algae is extensively used in aquaculture as the first feeding or first feed in the case of fish and shrimp larvae. And the genera which are used extensively happen to be Ketoceras, Skeletonema, Thalassiodesera, Isochrysis, Monochrysis, Tetraselmis, Chlorella, as well as Nanochloropsis. Let us have a look at the photographs of this Skeletonema, Thalassiodesera, Ketoceras, Tetraselmis, Isochrysis, Monochrysis, Chlorella and Nanochloropsis. This algae provide all the requisite level of protein, carbohydrate and lipids to the larvae. Apart from that, they also supply all essential minerals and vitamins. Now, what are the advantage of using the microalgae in rearing the fish and prawn larvae? As I said, it is the source of protein, and the size here is very, very small. The microalgae range from 5 to 25 micron. So, in the case of marine fishes, the mouth size is something like 50 micron. So, any food which is more than 50 micron cannot be ingested by this larvae, whereas this microalgae can be easily ingested by them. Then the microalgae stimulates enzymatic synthesis and onset of feeding in the young larvae. And again, microalgae are very important food for living live food organisms, and they have essential as an essential source in rearing of the stages of marine, estuarine and freshwater finfish as well as shellfish. And the microalgae, apart from serving as food, they also are important as conditioner of water because they strip off the nitrogenous substances so that the water quality is maintained. The next life food is happen to be the protozoans. You know that this is the basic or first group of animals, protozoans. Protozoans without any exoskeleton. It has got mostly the plasma membrane as the outer covering, and these protozoans are the nutrient capsules. Once when they go into the larvae or larval digestive tract, when they rupture, that means ready food, digested food is supplied to the larvae. So we have the marine protozoans as well as freshwater protozoans. From aquaculture point of view, the freshwater Infusoria, Paramecium and Stylonicus nixia is happen to be the important genera, while in the case of marine, it is Fabria and Eublotus are the genera which are important as live feed organisms. Now let us see these photographs of these species, which can be cultured and offered as live food for the fish larvae. You could see that this video is simply to show you what is exactly the size. It is about 400 times magnified, and you could see their movement also induces a kind of stimulus to the larvae so that they can easily feed upon this. As I said, because they are membranous, there is no difficulty in digesting this food material. Easily they can be digested. Now, what is the advantage of the protozoans? As I said again, it is very soft body and nutritionally very rich food. Again, their size is very small. Most of them happen to be 25 to 50 micron. Of course, bigger also is there. And this 25 micron to 50 micron size is ideally suitable for the marine fish larvae. They serve ideally as starter diet for the early stages of fish larvae. And also in a very, very cheap or cost effective conditions, we can have them cultured in high density. So required quantity of this protozoans can be cultured in a very simple, adopting very simple methods. Again, the movement of this protozoans is something which attracts predation by the fish larvae. Let us see this. Let us see the advantages of this protozoan. As I said, the movement is something which is very, very important, and even in a small aquarium, if you just take normal tap water and put 2 banana peelings and leave it at the window side for 3-4 days, easily you can see this protozoan. There is no need of any inoculum or any such thing. If you leave it for 3-4 days, there is a slimy layer formed on the top of the water, and there happen to be the protozoans, and with the proper sieving or staining, we can easily harvest them and feed them to the fishes. Let me pass on to the next life feed. The most important and most extensively used life feed. It happened to be the rotifers. Rotifers are commonly called as wheel animalcules, and these wheel animalcules are having very high diversity, and they are happen to be very tiny and most suitable life feed for aqua hatcheries. In that, one particular species, that is Brachioras is the one which is cultured in tons and tons and tons and supplied to all the hatcheries for satisfying the feed requirement of both the shrimp as well as the fishes. This Brachioras, there are 3 types. The first one is L type, which is fairly bigger one. It may have a size of around 400 to 600 micron meter. The second one is the S type, small type. It may have a size of about 150 to 200 micron meter. And the third one is the super small, which is the Brachioras rotendiformis. Even the S type is also with the rotendiformis. And this the small or super small size is about 100 micron. And in this species, there are quite a lot of again problems we are yet to solve. It is like the taxonomy of this and the reproduction patterns and their allocytes and what kind of mating behavior and mate recognition patterns. And if we just sort it out, we can try to make something where we can produce sexual bisexual reproduction. Also, a sexually reproductive produced animal will have better nutritive value than parthenogenetically growing animals. This is happened to be the photographs of the Brachioras Pericardis, which is, as I said, very extensively used life feed in aqua hatcheries. Apart from this single species, that is Brachioras Pericardis, there are quite a lot of other species. Rubens is there, then Caratella, there, many species are there, but we have not tried those species. There should be more and more studies on this species so that many more species can be used as life feed. Now let us see, this is the intensive culture of the Brachioras Pericardis, where you could see most of them are bearing the egg. So it is grown in captive condition with Saccharomyces cerevisiae as a feed and algae also as a feed, and it produces as much as every day every 4 hours interval or 5 hours interval one egg, and thereby in a very short time we can have a high density culture of these rotifers. And with regard to the advantage, as I said earlier, small body size and round shape, high reproduction rate and high density cultures, and they a slow swimming speed and habit of staying suspended in the water column, and they contain HUFA, that is highly unsaturated fatty acids, essential for the survival of a marine fish larvae. And suppose if a fish or a prawn requires any specific mineral or vitamin or any other compound, we can enrich this rotifers with that prior to offering to the fish, so that it also acts as a vehicle which carries the nutrient to the fish larvae. Again, continuing the advantages, it can supply at a food concentration required for the meeting larval metabolic demands and yielding high survival rate. That is, we can offer them in large quantities, even sometimes 500 individuals per ml. Enhance the digestive process of the predator, and different genetically distinct Brachioras Pericardis strains with different body size permits the larval rearing right from the endogenous feeding to exclusively exogenous feeding stage. Now let me pass on to the next group, Cladocerans, and this Cladocerans are commonly called as Waterfallias, and these Waterfallias are more common in freshwater systems than in estuaries and marine waters. And 4 Cladocerans species are important as fish food organisms, and they are Daphnia, Cymocephalus, Moina, and Cereodaphnia, and these are the very common genera. Many species belonging to this genera are important life feeds. This group, Cladocerans, again, most of the time they reproduce parthenogenetically. This parthenogenetic reproduction is an advantage because in a short duration we can build up high density cultures. Let us have a look at this Cladocerans. There is Daphnia similis, Moina macrocoba, Cereodaphnia, and these 3 happen to be the freshwater ones and very common. You cannot see a water body where these 3 are not present. So it is so common and so popular or abundant organisms. Whereas, let us have a look at this Podon, Evadne, Penelia, and these 3 happen to be marine species. And this marine species occurrence is not common. They are very seasonal, and that is the reason why we cannot apply them in aquaculture extensively, because their availability is seasonal and all the time we don't get them. But anyhow, recently from Southeast Asian countries, one species of Diaphanosoma, and specifically Celibensis, is identified in marine water. And this species is a common species, and this species again parthenogenetically we can culture them, and then high density culture is possible in a short duration, and this can be fed to the marine species, because the other freshwater species, they can tolerate only up to 6 ppt salinity. If it exceeds 6 ppt, then there is mass mortality of these organisms. Now let us see the video of the high density culture of this Cladocerans, which can be grown in with a very simple procedures. Let us see the advantages of the sclerocidins. They contain a broad spectrum of digestive enzymes. They have advantage of high reproductive rates. They tolerate a wide range of temperature and thrive very well on phytoplankton diet. And this can be used extensively in culturing the carps and other freshwater fishes. The next group is a living gold in the sea. It is copepods. Copepods happen to be the most numerous animals on earth. Their diversity and density is much higher than even the ants on the land. There's so much. And the marine realm, particularly the coastal regions, oceanic region, is harboring high density of these copepods. And these copepods serve as food for almost all, as it serves as a food for the larvae to the bigger mammals also, aquatic mammals also. And this nutritive value of this group is much higher than all the other foods we have seen earlier. Either it is rotifers or cladocidins or the other one animal, which is the artemia, which is extensively used. Compared to that, the protein content, the lipid content, more particularly the polyunsaturated fatty acids and essential amino acids are much higher in copepods. Among the copepods, there are 10 orders, but only 3 orders are free-living copepods. They are Calanoida, Cyclopoida and Herpacticoida. And in these 3 groups, there are quite a number of species which can serve as life feed. Among the calanoids, Acacia, Calanus, Pseudocalanus, Pseudodiaptomus are now extensively cultured. And in the case of cyclopoids, it is Ithona, Apocyclops and Thermocyclops, which have been tried as life feed. And herpacticoids, the Eutropina, Tisbe, Tigriopus and Onychocamptus. Now you could find here that calanoids are mostly pelagic in habit, while cyclopoids live in the column of the water, whereas the herpacticoids are mostly benthic. So this group offers themselves as a feed for the fish larvae which are pelagic or which are column dwelling or benthic. Now you just see the photograph of some of the calanoid copepods, and this group ranges 2 to 5 millimeter in length, and they are the most dominant in the marine habitat. This group is more predominant than the cyclopoids and herpacticoids. Then the first rows happen to be the cyclopoids. We can easily distinguish them through their morphological features. In the case of calanoids, they will carry a single ovisac, but in the case of cyclopoids, it will carry 2 ovisacs. And the third is the herpacticoid, where it will carry only 1, but the shape of the herpacticoid is barrel-shaped, whereas the cyclopoids and calanoids are distinctly different. So these copepods are something which is very vital for developing the marine fin fish larval production, because they are sexually reproducing, and if you just try to follow their development, they have themselves in their life cycle 12 larval stages, 6 naupliar and 6 copepodate stages. Let us see the cyclopoid high density culture which has been done by us in our lab. Now, as I said, these are happen to be the naupliar 1, naupliar 2, naupliar 3, naupliar 4, naupliar 5, naupliar 6. Sometimes in the case of the herpacticoids, this naupliar size is 30 to 40 micron, or it may be around 30 to 50 micron, and it can go up to even 200 micron when it comes to naupliar 6. What I mean to say here is, this size of this naupliar, 40 micron, 50 micron, is highly suitable for the first feeding of the fish larvae, apart from the microalgae. And this feed is also highly suitable. And in fact, in nature, when we try to just collect the fish larvae from the nature and analyze their gut content, and their gut contains mostly the copepod naupliar as a feed. That means this is happen to be the natural feed for the fish larvae. Now you just try to have a look at the SCM pictures of the copepod naupliar. That these pictures we have specifically taken, I have included, because sometimes some people have got the apprehension that these larval forms may have some kind of spines or anything if we just try to offer them to the fish larvae, because the yolk sac and the body itself is membranous or very formative stage, and the skin is not fully formed, digestive system is not fully formed, then these spines of these copepods may rupture them or puncture them or cause some kind of damage. But scanning electron microscopic pictures clearly shows there is nothing of that kind in many of the species which are offered as live feed. Again, as I said, those are happen to be the 6 naupliar stages, and these are happen to be the copepodate 1, 2, and then these happen to be the copepodate 3 and 4, and then this is the female and the male, and the male and female is distinctly different. Now this is a very interesting aspect where myobenthic herpacticoids. I was telling you with regard to the calanoids, cyclopoids and herpacticoids. I said that calanoids are pelagic, cyclopoids, of course, they can swim in the column water, then herpacticoids are benthic. There are herpacticoids which are myobenthic also, that is, they exist in soil sediments and they live in the interstitial water. Based on their life in the interstitial water, in the sand sediments, their body structure is modified. Anyhow, their size is highly reduced. In the case of the benthic herpacticoid, the size may be 1 millimeter to 2 millimeters. But here the maximum size is only 700 micron. You see that this particular animal, this itself is 700 micron, but is having a egg sac holding some 5, 7, 10 eggs, and this egg may be less than 20 micron. Likewise, you could see the egg sac. Here on egg also in the developmental stage you can see. So you could see sometimes in the case of herpacticoids, the size of the egg is so small, and this eggs can be a good feed for the fish larvae. But at the very same time, 20 micron, 30 micron gain a very nutritive food at a proper size. If we provide such food, then the development, growth and survival of the fish larvae will be much better. You can produce quality seeds with that. So again, considering the advantage of coffee pods, we need to have more and more studies, and because Rotifers and Artemia are readily available, we are using it. We have not developed a technology for the production of the coffee pods. It is the need of the hour, and as it is, as I said again, it is happen to be a natural feed for the larvae of juveniles of many fishes and crustaceans. They have wide spectrum of size, as I said, from 40 micron to 1000 micrometer, and many marine fish larvae feed on copypods, egg and naupliae during their first few weeks of life, and a number of marine larvae fish cannot be reared, or we cannot grow them to the juvenile stage without copypods. We cannot raise them using either Rotifers or Artemia. So we have seen some of the indigenous live feed, and this live feed, as I said, is very, very important for the production of the young ones of marine fishes as well as the shrimps. Apart from that, this live feed play a very, very vital role in maintaining the productivity, because they feed on the primary producers and in turn transfer the energy to the secondary consumers. And many of them are also indicators of the pollution, and many of them are involved in bioremediation of pollution. So this group is vital from many particular aspects, and if your system has got a rich diversity and abundance of this organism, that means the water is stable and the system is stable, the ecosystem is good, and there is good productivity. Once when we have such a kind of a stable ecosystem, then human beings can draw many attributes from this system.